Hello, my name is Natalie Arias, and here today I will be talking to you about methanogenic archaea and the ruminant. And this will be my second project for my master's thesis. Ruminants are exceptional organisms in that they have an incredibly abundant microflora with different varieties of microorganisms that take place and inhabit their gastrointestinal tract, all the way from the esophagus and different parts of their four-chambered stomach to the small and large intestines and even further down. They have plenty of different types of organisms, including eukaryotes, which are multicellular, and the most predominant species are anaerobic fungi and protozoa. They also contain a variety of bacteriophages and viruses. And lastly, what I would like to focus on in this presentation is a specific form of prokaryotes, or a specific form of unicellular organisms. We've talked in great depth about anaerobic bacteria and the help that they do in degrading feedstuffs so that they can produce energy compounds, but we will also be talking about methanogenic archaea in this presentation. And here in the middle, I have some pictures of what these methanogenic archaea could look like under the microscope. So what exactly are methanogenic archaea? Well, they're simply referred to as methanogens in literature, and they are the most prominent archaeon in ruminants. Some say that this is the only type of archaeon inside of ruminants, but others have different proportions that they have discovered. So we don't really know for sure what is their average abundance inside of a ruminant. Methanogens utilize excess hydrogen gas and carbon-containing compounds to produce methane. And here on the bottom and on the right, this is a graphic that I've shown you before, where a ruminant such as this lovely Highland cow eats feed carbohydrates, which then get broken down into glucose by different microorganisms that are amylolytic or starch degrading. Then this glucose goes to a variety of pathways producing pyruvate via glycolysis and even releasing multiple compounds of hydrogen gas. The methanogens are a hydrogen sink, and so they will use this excess hydrogen to produce methane and release that out of the system. And this is only, as I said previously, one of the ways in which hydrogen can be used in the ruminant's body. These are two slides that I took from Dr. Calloway's slide deck as we previously saw them in a class. And I wanted to include these because these really showcase what exactly the methanogens are doing and some various names of certain methanogens that are highly prevalent inside of the rumen. So as you can see on the left, formate, which is a one carbon containing compound, get broken down by methanogens to produce methane in one step. However, formate can also go through a two-step process to produce methane in conjunction with other bugs or other microorganisms. So formate can be broken down to hydrogen gas and carbon dioxide, and then these two compounds can be used by methanogens to produce methane. In addition, on the right-hand side, we have a phenomenon called interspecies hydrogen transfer, and this is a phenomenon in which archaea will cling to the sides of other microorganisms in order to absorb the hydrogen from what they are using. And so some common genus and species of this archaea, this methanogenic archaea, include Methanobrevibacter ruminantium. That would probably be the most abundant one that I've seen in literature, and also these others that you can see on the slide. Now here for a little bit, I would like to talk about the details on how exactly do we isolate archaeal DNA, as it's a process that can look very similar to isolating bacterial DNA, but there are some differences that actually make this process really, really interesting. So we'll go over how do we sample fluid from the rumen, as well as how do we extract this methanogenic DNA. Even though this topic may seem a little elementary, it's one of my favorites within ruminant research and just talking about the ruminants is how we collect samples differently, and how can that affect our results? How can that affect how we interpret these results? And so on the left-hand side, you'll see me collecting ruminal fluid via a cannula or a fistula. This technique is called rumen cannulation, where these animals are surgically implanted with a rubber stopper in order that you may be able to stick your hand in, then remove some of the top levels of the rumen fluid closest to the hole, essentially, because it's more exposed to oxygen being so close to the rubber stopper. Then inside you can feel the papillae, you can feel all the different hay material and the fluid material, and you can even feel the muscular contractions within the rumen. So it's really awesome to be able to go to the farm and collect. The advantages of rumen cannulation is that it is very easy sampling. You simply stick your hand in and you take the material out and you guard it in a 39°C thermos in order to keep that temperature the same between when you removed it from the rumen and when you go to the lab. However, this is a very expensive process as it takes a surgery to be able to put the cannula in the animal. It also is quite invasive, and you have to monitor the animal and make sure that this cannula is not being infected or anything like that. And with how expensive and invasive this procedure is, oftentimes there are fewer animals on the farm that have this cannula versus non-cannulated animals. So what many researchers, including what our group has done in the past, is turn to esophageal tubing or simply stomach tubing. This is definitely a more cost-effective way. All you have to do is buy certain equipment and then you're good to go. And I'll briefly explain how we do this. So Michaela, who is the girl in the back, she is putting a tube down this steer's throat, and coating the tube is a metal pipe so that while we are extracting liquid from their rumen, the pipe is not going to be destroyed by the chewing sensation that the steer automatically inherently has. So she is keeping this tube very steady within the rumen, sometimes even being jolted around by the animal as he gets kind of uncomfortable. And I'm here on the bottom, and I am holding this collection tube so that the pump can then pump liquid out of this collection tube and into the vat that I am holding. And so frequently I'll have to open it up to release pressure, but ultimately I'll fill this up and hand it to someone else to pour into a 39°C thermos, just like we did with the rumen cannulation method. So with this technique, even though it is quite stressful for the animal and we can't sample as many times in a day as we could with, say, rumen cannulation, more animals can be sampled per time. And so it is stressful, but it is oftentimes more powerful for statistics. Another downside of esophageal tubing is that more personnel are required. As you can see, I'm the only one pictured in the left image, but we have Michaela and myself in the second image here. And a lot of times there is contamination with saliva, and that can even lead to skewed results, and you don't even know why your experiment failed. Well, saliva could be an option for why there was very little microbial growth within the rumen sample that you could be testing. So I have a question for you, just to think about these two sampling types. Which technique, based on each of these options, would you choose for a future experiment? Here's a slide fully dedicated to DNA extraction. On the left-hand side, you can see a picture of our lab in the middle of this process, and we have a lot of different tube holders because it is a very time-consuming process depending on the number of samples that you have taken from the farm and you've prepared. We're going to go over some umbrella terms with DNA extraction and the different types that you can have. The first, being enzymatic, which uses a lysis buffer and different chemicals in order to lyse and break apart the cells. Additionally, you have mechanical DNA extraction. So the physical effects of this is using bead beating tubes, where beads at a super fast rate shake up and disrupt the cell membrane in order to release the nucleus and the genetic material out. And one study by Yu and Morrison in 2004 has been cited over 1,200 times for its novel and effective way to lyse cells using a bead beating tube plus a column method. Additionally, there are commercially available kits that just contain everything that you need to have, and they specialize in different types of microbial DNA extraction, such as from rumen samples, fecal samples, buccal samples, and the like. In this picture at the top, you can see that there is a particular fashion that DNA extraction happens, and it may vary by different protocols, but this is the general formula. You'll spin down everything that is waste into this number 2 slide, and that gets washed away. And only the DNA theoretically is left, and then you can finally spin that again and elute the DNA properly. That's all that you need to know for DNA extraction. And as a tidbit, our lab does a mixture of enzymatic and mechanical extraction. Now I'd like to touch on the phylogenetic marker genes that estimate methanogenic abundance and CH4 production. And this is extremely important and vital when we are moving on past the DNA extraction phase. Now, what genes can be sequenced? Well, on the left-hand side, we just talked about this in class. There's a gene called methyl coenzyme M reductase subunit alpha. Now, this encodes an enzyme, MCR, that catalyzes the final step of producing methane. And it is hopefully innate, but it is unique to methanogenic archaea. So this gene helps to encode this enzyme that'll move the final step forward in producing methane as a byproduct. So as you can see below, this is the reaction that is catalyzed by the MCR enzyme, and you can see that the methane is being released in the side. Now we have our classic 16S rRNA gene, or simply RRS, or you may even see it labeled 16S rDNA, or ribosomal DNA. Now this gene encodes the RNA of the small subunit in a prokaryotic ribosome. Here on the bottom I have a picture of a ribosome in case you have forgotten what the structure looks like. And on the bottom right, you can see the exact structure of the gene. It's quite large, has a lot of kinks to it, and it is conserved between bacteria and archaea species. So between two species of bacteria, you'll have conservation of certain portions of the gene, and also between two species of archaea, you may have similar portions conserved between those two species. So you can use these conserved regions, C1 through C10, in order to design primers that can be used during a future process called PCR, and that will amplify a portion of the gene that you really want to look at. Additionally, you can use the hypervariable regions, V1 through V9, to design further primers for PCR and amplify those really species-specific regions. So on the bottom right, you can see a long chain of C1, V1, C2, V2, and so on. These are the interspersed regions that are conserved and variable, and that aligns with their letters. So there you have it. We have different popular species or different popular regions that can be highlighted depending on the species and depending on the kind of work that you want to do in sequencing a particular bacterial or archaeal species genome. Now, most microorganisms cannot be cultured because of the difficulty to isolate and create a viable medium, and so researchers have looked to other ways to be able to study the microbiome. Enter in Sanger sequencing, which began the conversation in order to elucidate the sequence of different species of bacteria and different prokaryotes. But next generation sequencing took sequencing to the next level. So we can do 16S rRNA gene sequencing via the Illumina MiSeq, which Illumina specializes in short read sequences, whereas other companies such as PacBio and Oxford Nanopore may specialize in long read sequences. Additionally, this next generation sequencing is open to both bacteria and archaeal DNA and RNA, which is perfect for our case when trying to elucidate different methanogenic sequences. As I said previously, we can use different hypervariable regions for these PCR amplicons and primers. Popular ones include the V1 to V3 region, also the V4 to V5 region, and as you can remember, our 16S rRNA gene contains 9 hypervariable interspace regions. When we pair next-generation sequencing with bioinformatics, the data can be unstoppable, and we can really find a lot of cool research that really determines what kinds of bacteria are populating this microbiota within the rumen or within feces. On the right-hand side, you'll see a process of how we go from next-generation sequencing all the way down to visualizing what we have, and we use different bioinformatics software. One that is very popular, especially here in our animal science department, is QIIME, spelled QIIME, and that really does take a lot of the trims and a lot of the things that do not belong within the DNA sequence and isolates it down to what is most important to focus on. There are even more ways today to be able to quantify abundance and identify rumen microbes, and I've included a little tidbit of some other analyses on the right-hand side, including phylogenetic array, shotgun sequencing, RNA sequencing, and reverse transcriptase, qPCR. But the main one that I want to focus on here is quantitative real-time polymerase chain reaction, or simply qPCR. This is a method that functions very similarly to how PCR works. In normal PCR, we just amplify the particular gene region that we're trying to look at using different primers to hook onto those sequences and allow the polymerase to amplify those sequences even more, basically make millions of copies of a particular sequence in order for our sequencer to be able to read it and detect a signal. But qPCR takes it one step further, and we can actually quantify what DNA we have and how much of it within the same instrument that we amplify it. This is amazing because we can use not only our two genes that we learned about, RRS and MCRA, but we can do so for a variety of domains, prokaryotes and eukaryotes. We can also use what's called universal and species-specific primers. So, for example, if you're just looking for the presence of anything within the domain of Archaea, you would use a particular universal primer that looks at Archaea. But if you wanted to isolate a specific species, such as Methanobrevibac ruminantium, then you would find a primer within past literature to isolate, amplify, and quantify just the region of MCRA, for example, from that species. Now, qPCR is extremely powerful, but because of its novelty in this domain, in this region of research, there's weak cross-study comparison. So methods are not typically the same, primers used are not typically the same, and there's just a small known database which can create bias when trying to interpret and display results. So that would be the downside to performing qPCR, but it is a very novel area of research in terms of defining the methanogenic profile of a ruminant, and so that is what I want to do within my 2 years here of my master's. We've spent a lot of time in this presentation discussing what we can do after we have observed the archaeal DNA and quantified it. But what if researchers want to go about this in a less invasive way? Instead of sampling directly from the rumen via a cannula or via esophageal tubing, are there other ways to be able to quantify archaea? Well, research shows that we can look in the feces, in the urine, and in the milk. Let's start with the feces. So it has been studied that in feces of ruminants, there is a compound that is not digestible in the gastrointestinal tract and then escapes in the feces, and that is called archaeol. Archaeol is the archaeal membrane made of this dialkyl glycerol ether bilayers. This is different from other types of cellular membranes, and so it is easy to collect and isolate and pool together. This is a very interesting and esoteric level of research, so once again, not a lot of cross-study comparison that can happen here. In the urine, we can look at this purine derivative called allantoin, which is excreted through the urine and contains a lot of nitrogen, to be able to predict microbial protein flow from the rumen. Now, this idea sounds excellent, but there's a lot of assumptions made about this purine flow out of the rumen and into the urine, such as, does this purine derivative allantoin get degraded at all? Is it still the same as what it was in the rumen? And also, are there other purines that are being lost that we just cannot quantify because they go to other pathways within the ruminant? So lots of questions up there. And then finally, we have milk VFAs that can be run through a VFA process of analysis. Additionally, we can look specifically at odd and branched chain fatty acids such as isobutyrate and isovalerate, and those can be used to correlate to the ruminal volatile fatty acid ratios. So what goes out of the milk could potentially display what is being contained within the rumen. Additionally, we have mid-IR reflectance spectra, which has been used to predict the individual methane emissions. So this is a very early on technique that needs a lot more research done and a lot more studies, but if IR can be used to predict other sorts of gases, could it also be used to predict methane from the rumen? Again, weak correlations demonstrated because this is such a new area of research that statistics are not super powerful yet when it comes to testing this, but all in all, the whole reason why I'm bringing this up to you, these non-invasive techniques, is because grazing animals are prevalent across the world in different developing countries and different areas of agriculture. They produce more methane than grain-fed animals, and so being able to quantify methane production and other key players that produce this methane, namely the methanogens, is so important. And it could be more cost effective to be able to isolate feces or urine or milk than to go in and establish a cannula or to get the labor in order to process samples that were collected via tubing. So these are just different techniques that can be tried to isolate methanogens. The last thing that I want to say about the relationship between methanogens and nutrition is all boiling down to this question here in red. Can a more developed methanogenic profile help us to understand nutritional phenotypes such as CH4 emissions, feed degradation, and even feed efficiency? Well, two papers noted in the review that I'm going to highlight on my last slide is this. Gill et al. In 2011 found a positive relationship between forage-fed steers and fecal archaeol, or the fecal matter of archaeal membrane, that dialkylglycerol ether that is so unique to Archaea. Additionally, there was a positive relationship, not as strong but still there, between forage-fed steers and methane production on a dry matter intake basis. So those are great stats. I mean, that's great to know that there is a correlation between eating more forage and this release of the fecal archaeal membrane content. McCartney in 2012 found a ruminal selective retention which is very unique to containing the methanogens within the rumen, and this is quantified potentially by the methane to archaeal ratio. So a higher ratio could indicate that there is more methane being released and a higher retention of these methanogens within the rumen. However, this could be skewed results as the stage of lactation did play a significant role in these results. As you can see, this p-value of less than 0.005, excuse me. So there are a lot more questions that we have than we have answers to in regards to the methanogens' impact on nutrition, on what comes out of these cows after they are done feeding, and so it's important to answer these. I've included here where I got my pictures and my references, and I'd invite you to take a look at the review that I've highlighted here at the top. The top two are reviews, and they were very helpful in helping me understand more about methanogens and their role in the ruminant. So thank you very much for your time, and I hope to see you soon.